BlogTalk Radio.
Welcome to the AME Food Testing Show. Today's guest is Dr. Lee-Ann Jaykus, Ph.D., Professor, Food Science, North Carolina State University, Raleigh, North Carolina. Our topic is norovirus, a foodborne pathogen. Dr. Jaykus's background academically has been a bachelor's degree from Purdue University, a master's degree from Purdue University, and a Ph.D. from the University of North Carolina at Chapel Hill. Her research activities have focused on application of molecular biological methods for the detection of pathogenic microorganisms in foods. Her current research projects involve the development of nucleic acid amplification technology for the detection of human enteric viruses, human enteroviruses, hepatitis A virus, Norwalk virus in shellfish, fresh produce, and ready-to-eat food commodities. Her additional research opportunities exist for developing similar methods for the detection of Listeria, monocytogenes, and Salmonella from dairy products, with specific focuses on bacterial concentration and refining of molecular methods to facilitate the real-time detection of foodborne pathogens. Her group is also actively involved in the application of quantitative risk assessment methods for the evaluation of public health risks of foodborne pathogens. Dr. Jaykus, would you like to update us on any other of your current projects? And I'll now invite her to speak with us. Dr. Jaykus?
Hi, this is Lee-Ann. Just one additional project relative to viruses. Our group heads up what is called the USDA NIFA. NIFA stands for National Institute of Food and Agriculture Food Virology Collaborative. We also call it NoroCore, N-O-R-O, capital C, capital O, capital R, capital E, and that stands for Norovirus Collaborative for Outreach, Research, and Extension. NoroCore is a large project that's funded by the United States Department of Agriculture that really seeks to better understand the food virology problem, really from using a farm-to-fork approach, and also tackling it from a research perspective, an educational perspective, and an outreach extension perspective.
Well, Dr. Jaykus, welcome to the show. We'd like to begin with a general discussion on what is a virus.
Most people in food safety are certainly used to hearing about bacterial agents like Salmonella and Listeria monocytogenes, and viruses are actually a very different type of microorganism. Unlike bacteria, they are not, quote, alive. They are called obligate intracellular parasites, which means that they require a live host cell in order to replicate. Now, they can't replicate on their own without that host cell. And in the case of the human enteric viruses, which are what the foodborne viruses are, they are specific for human cells, in most instances specific for cells in the gastrointestinal tract. So they're unique from bacteria in that they're much simpler in structure. Most of the viruses we work with are very small. They're about anywhere between 25 and 40 nanometers in diameter. They have a protein coat, and inside of that protein coat is a nucleic acid molecule. Usually it's an RNA molecule, which is the genetic material for the virus. And that's all they are. That's all they consist of. And so what they do is they'll infect the host cell. They release their genetic material into the host cell, and that takes over the machinery of the host cell and causes the host cell to die, to lyse, and then to pop out new virus particles. So they're kind of very tricky in the way that they cause infection. Another thing that's important to recognize is that because they can't grow outside of a live host when they're in a food system or in the environment, they can't replicate. So unlike bacteria that can multiply in the environment or in food, viruses can't do that. But at the same time, they also can persist in very severe environments for very long periods of time.
Excellent. In our discussion, what is a norovirus?
Noroviruses are a group of viruses. They fall in the family called Caliciviridae, and calici means cup-like, or calici comes from the Greek chalice. So if you look at these viruses under an electron microscope, they have these cup-like indentations on their surface or on their protein coat. And we now know that there are several genera or groups of caliciviruses that are associated with disease in all kinds of animals. But the noroviruses are one of those groups, and they cause they cause acute gastroenteritis in humans. And we now know that this particular group of viruses are the leading cause of acute gastroenteritis worldwide, and they're also the leading cause of foodborne illness.
You and I were chatting earlier about the CDC's estimate of known causes of foodborne illnesses. I was shocked to see that 51% was accounted for norovirus and only 38% for bacteria.
That's correct. There was pretty much a landmark paper that came out in 1989 by Paul Mead that was sort of the beginning of this recognition that viruses were a significant cause of foodborne disease. The paper that you're referring to was by Elaine Scallon, and it's more updated data, but still we believe pretty strongly that about half of foodborne illness is caused by noroviruses. And keep in mind that's foodborne illness of known etiology, and actually if you take into account foodborne illness of unknown etiology, for which the numbers are much larger, probably some, if not a significant proportion of those diseases are also caused by some sort of a viral agent.
Well, as we look at that and address the issue, can norovirus be a foodborne pathogen? Your obvious response would be yes.
Yes.
So what are the detection methodologies to look for norovirus?
Well, I guess you have to look at detection technologies as sort of what might be currently available versus what's in developmental phases. I think you kind of have to take a step back, at least from the type of testing that's done for very well-characterized bacterial agents of foodborne disease, because we don't have anything like that for noroviruses. So let's look first at clinical samples. So if you're going to diagnose or try to diagnose a norovirus infection in a person who's sick, there's a couple of methods that can be used. Certainly an antibody-based method has been used by some, but in most instances a molecular method, specifically something called reverse transcription polymerase chain reaction, which is basically a DNA copy type of method, is used. Now, there's a couple things to keep in mind. The first is that we don't, at least in the United States, we don't have a licensed clinical detection method that can be used for routine diagnostics in the population. So I can't go to the doctor and say, "Can you test me for norovirus?" We do have a method that can be used in outbreak situations, but not for individual diagnosis. So from a clinical standpoint, it's very difficult to diagnose the disease in the community. Now, most state public health labs and certainly the CDC routinely use reverse transcription polymerase chain reaction, or RT-PCR, for diagnosis. But but that, again, is almost always in an outbreak situation. When you get to food samples or environmental samples, it becomes a lot more complicated. Firstly, because these viruses can't grow outside of a host, and actually we don't have a method to grow noroviruses or human noroviruses outside of humans. So we can't use the traditional cultural enrichment methods that most people use for detecting bacterial pathogens in foods and environmental samples. So that's out. That's not an option. Another big thing to consider is that because the viruses have such a low infectious dose, and because contamination in foods might well, be at very low levels, you might expect a product that is contaminated to only have a few virus particles. So you're sort of faced with the same issues you have with bacterial detection. You have not a lot of contamination there, but at the same time you can't do an enrichment step. So generally what we do with these types of samples is we concentrate and purify the viruses from the sample matrix. So you might take a 25-gram sample of strawberries, for example, and concentrate and purify the virus from that sample into a much smaller volume, say a milliliter, which is, for those who don't work in the lab, is a pretty small volume. And then we process that concentrate for isolation of the nucleic acid and for RT-PCR for detection. So that's the method by which we normally do this type of work.
So for an environmental sample, for example, in a food production plant where potentially the virus has contaminated a work area or food contact surface by a worker who has been a host, would a swab be collected and then perhaps sent off to a lab?
Yeah, a swab could be collected, and actually in the scientific literature there are many, many, many examples, particularly in association with foodborne disease outbreaks or propagated outbreaks on a cruise ship, for example, where investigators do go in and they take a swab of surfaces and they send them into the lab, and then that swab is processed for testing using molecular methods.
So the question is, what are the detection methodologies? And so there are very few options open to food production managers that would like to examine their production facility for this particular type of organism.
Yes, I would say, there are some companies that are sort of in developmental phases for potential kits, for lack of a better word, that you might use for detection of noroviruses in a food sample, an environmental sample, in swabs, etc. It's a very complicated. The development of those kits is complicated by a lot of different factors that probably are sort of beyond the scope of this particular show. And there is one company called Ceram, I think it's spelled CERAAM, in France, that does sell a diagnostic kit for noroviruses as applied to certain foods and environmental samples. So there is something out there marketed. But none of these have really gone through formal approval processes yet. And the difference between actually applying the molecular amplification method, which it's relatively easy to put that into a kit format and make it pretty user-friendly. The bigger issue, or the more complicated issue, is how do you treat that sample to promote the recovery of the virus before you go into the detection platform or the amplification step. And that's kind of the sticky wicket. That's what is so difficult to do, requires a lot of technical expertise, and is very difficult to actually put in some sort of a kit format.
So aside from the sample preparation issues, is it possible for a food production manager to physically look at a worker and make some sort of qualitative determination, hey, this person shouldn't be touching my product or be handling my product?
Well, that's a great question. Any worker who has active diarrheal disease or certainly is vomiting on the premises, or has even complained about vomiting, 24 hours before coming to work, should be highly suspect for a norovirus infection. And really, in my opinion, should be excluded from work. It becomes a lot more complicated with some of the emerging evidence that's coming out really within the last couple of years, that people can shed virus pre-symptomatically. They can shed virus post-symptomatically, so after their symptoms have resolved, they can shed virus for many, 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 many days. And there's even a bit of emerging evidence, although we need to do some more confirmatory work on this, is that there's some people who actually are even shedding virus in their stool without showing any symptoms at all. And so if you're in if you're in the business of manufacturing food, particularly in the retail setting or a setting where you have a lot of hands-on contact with food, managing those types of situations become very, very complicated.
So, Dr. Jaykus, how are the virus organisms spread? You mentioned the word shedding. That's interesting because most people feel in food production, if you wash your hands, place gloves, that there shouldn't be any issue.
There really, we now know there are many ways that noroviruses are spread in a community. Okay, so we talk about that as transmission. Okay, and foods is only one of those transmission routes. So you can get a norovirus infection by close personal contact with somebody who's infected. You can get it from drinking contaminated water. In a daycare center, for example, you could have some virus on a ball or on a surface and touch it, and then put your hands in your mouth and get infected. So there's a lot of different ways that you can actually you can come in contact with the virus to cause infection. But the two sources of virus contamination really are stool or fecal material from an infected individual, and also vomitus from an infected individual. So, for example, if an infected individual is vomiting, and vomiting is the hallmark symptom of norovirus infection, and so well over 50% of people with a norovirus infection will vomit. So there is a lot of evidence that the virus is actually shed or released in a vomiting incident, and probably in very, very high numbers. So the classic fecal contamination route, those of us in food safety know all about. People get fecal material on their hands, and they don't adequately wash their hands, and they go on to touch food, and the food becomes contaminated. So certainly, certainly adequate hand hygiene is incredibly important, and gloving can be important as well. The issue there is that if you look at the literature on compliance with hand hygiene, it's really abysmal. And so, we can educate people and even do some observation to try to assure that hand hygiene measures are met. But when you actually look at what people are doing, a lot of times they're not doing what they're supposed to be doing when it comes to hand hygiene. Another sort of complicated factor, not so much in food service because this isn't state of practice in food service, but certainly in in the healthcare setting, is that the alcohol-based hand sanitizers that we all see out there and see people using all the time are not good at inactivating noroviruses. So you can't really use alcohol-based hand rubs as a means by which to assure that somebody's hands are going to be virus-free, at least when it comes to noroviruses. So that adds another layer of complexity. You don't want people getting a false sense of security that, I used a hand sanitizer, so I don't have norovirus on my hands. Vomiting incidents, on the other hand, are extremely complicated, very poorly characterized. It's actually an area that my group is working on really extensively right now. So we don't know. We believe that when an individual vomits that there's some degree of aerosolization, and that aerosolization can reach people at least within a certain radius of the vomiting incident. The other thing is that, of course, surfaces that become contaminated in a vomiting incident, whether that's directly from vomitus or through aerosolization of vomitus, will end up with some virus deposited on those surfaces. And there's a lot of evidence, both laboratory evidence as well as epidemiological evidence, that those viruses that become deposited on surfaces are, number one, very persistent, can persist in the environment for weeks at a time, and number two, are highly, highly resistant to disinfection. So in many instances, some of the CDC recommendations are using chlorine concentrations as high as 1,000 parts per million on a surface to inactivate any potential norovirus contamination. Well, those of us who work in food service or in food safety know that 1,000 parts per million is a huge quantity of chlorine, for many surfaces, it's not practical, and it may not even be allowable from a regulatory standpoint.
Well, those offer a number of challenges. The one that comes to my mind is the aerosolization capability of this organism, and the other issue is we can't kill it, is what you're saying, very easily.
Correct. The aerosolization, as I said, we're in the process of doing a lot of experiments to try to characterize that. They're kind of creative ways. We're working with some engineers in those projects, and to try to better understand the degree of aerosolization, perhaps how far the virus might be deposited in a vomiting by the process of vomiting. And the idea with respect to that is if you can better understand those factors, you can build risk models that will enable you to understand the dynamics of virus spread and potentially come up with some recommendations on how to manage vomiting incidents in a retail environment or in a home or school or really wherever. So that's a huge area of need that our NoroCore group is working on. This other issue of how do you inactivate them? There's a lot of interest in developing novel methods novel virucidal methods for hand sanitation and also for surface sanitation. That has its own complexities, not the least of which is issues associated with being able to license those products and make virucidal claims. And that's sort of a regulatory issue, but that is something of a limiting factor in the ability of industry to potentially respond to the need for better disinfectants and sanitizers.
Very good. Let's address for just a few minutes, since the principal audience of this show are food producing managers, food quality managers, food safety managers, and food security managers. What can those types of individuals do to screen or potentially protect their end users from norovirus?
Well, right now there's not a whole lot that they can do in terms of screening. In the absence of a clinical diagnostic or in the absence of really well-validated diagnostics, even for foods and environmental samples, it's hard to screen. And, I'll put a plug in from my perspective, testing is one part of a quality assurance program, but it should never be the centerpiece of that program. So, I think testing for viral contamination may have a role when the methodology is enhanced, but it should never be relied on as sort of the be all and end all in your control program. I think a lot. I would say it sort of depends on what your unit operations are. Let's talk a little bit about the produce industry, because you mentioned that the produce industry was an important constituent for you. I would say that it's important for the produce industry to be aware of the potential for irrigation water to be associated with contamination. So certainly being aware that, or aware is not the right word, but certainly trying to assure that irrigation water has not been contaminated with human fecal matter, I think is very important. Also managing people in the field who might be picking produce, so attention to hand hygiene, making sure there's adequate sanitizers in latrines and hand washing stations in the fields. One area that has certainly been documented as a means by which product can become contaminated with virus is small children or even adults defecating in the field. So, you need to pay attention to make sure that that's not happening. Another issue is potential for gloving, although you have to make sure people don't use the restroom while they're, while they have gloves on. Another aspect, I think, is really assuring that you have good and ongoing education on hand sanitizing and hand hygiene in your plant. And then observation is key. I mean, you have to be aware of the health status of your employees, and you have to promote, policies that allow them to be honest about, being out of work when they truly are sick with something that is going to potentially, or has the potential to result in a major food safety problem.
Those are excellent recommendations, Dr. Jaykus. In my food consultation, I generally ask the food safety manager to visually look at and screen the workers for various things, such as hand hygiene, nails that are attached or glued to their fingernails, and other types of hand washing type protocols. What other suggestions might you offer to these managers as they are looking at their employees prior to working? I'm particularly concerned because I eat out quite a bit, and I look at these food workers, and I'm concerned many times.
Well, there is quite a bit of evidence out there that it's very hard to adequately clean your hands, okay, that people don't understand how to wash hands. And if they do sort of a slipshod job at it. From my perspective, even looking at hands and scrutinizing nails is really important. I mean, fecal matter can get stuck underneath nails very easily, and if you're not scrubbing those nail beds or underneath the nails, it stays there. And of course, these viruses are very, very persistent, so they're gonna stay there as well. So I think certainly that kind of attention to the extent that you can do it without invading somebody's personal space is important. But I also think that this whole issue of kind of knowing your people and knowing I guess almost intuiting if they seem off or if there's something, they just don't look like they feel well. I mean, I think that that's also another way that you can, potentially catch something before it becomes a problem. And I mean, the industry is notorious, and I understand the reason for this, notorious for not paying people sick leave. And so some companies that, the absence of a sick leave policy really kind of hinders the ability to manage diseases like norovirus, because people kind of bear with it and they work through it, and they may not even understand how they could maybe even unintentionally be promoting a contamination incident.
Very good. Dr. Jaykus, you've been an excellent interviewee today. I'd like, if you could, is to update us on your research and your projects. I understand you recently received a $25 million grant to help nullify norovirus.
Yes, that's our NoroCore project that I was talking a little bit about at the beginning of the show. So the purpose of that project is to ultimately to try to reduce the burden of foodborne illness that is associated with human enteric viruses, particularly the noroviruses. We have divided the project. It's a very big project, so I lead the project, but we actually have 16 or 17 other institutions that are involved in the project, including representatives from USDA as well as the FDA. And we've divided our work into what we call 6 core activities. 4 of those core activities are largely research, and the first core is molecular virology. And in that core, we're looking at potentially developing a human norovirus strain that we can culture, which would make it much easier for those of us who work in this area to actually work with these viruses. There are other initiatives there, but that's probably the biggest initiative in the molecular virology core. We have a detection core where we're seeking to produce better detection technologies for noroviruses, both in clinical specimens as well as in foods and environmental samples. And the ultimate goal in that regard is to develop some candidate methods, but also to work with commercial partners to try to bring these potentially to commercial fruition. The third core is epidemiology and risk analysis, and the whole purpose here is to better understand the burden of foodborne disease associated with human noroviruses, both from an epidemic standpoint as well as from an endemic standpoint. So how much disease are we actually seeing sort of circulating in the population as a baseline, and how much of that is associated with foodborne routes. We're also in that core looking at the economic costs of norovirus infection, as well as developing risk models to help identify critical control points and management strategies to reduce the burden of disease. The fourth research core is prevention and control, and we're looking at basically working pretty broadly with the industry to try to identify ways to prevent noroviruses from entering the food chain, or if they're in the food chain, find novel ways to destroy them. And that can be anywhere from processing technologies for certain key foods up to the development of better sanitizers, better disinfectants for hands, things like that. So those are our 4 research cores. We then have an extension and outreach core that focuses on the 3 major food sectors that are interested or concerned about noroviruses, and those are the mollusk and shellfish industry, the fresh produce industry, and the retail and food service industry. And we have a variety of projects where we're working with companies and stakeholders to do things like get the message out about noroviruses and develop better strategies to prevent contamination in those particular products within the confines of the production, processing, preparation scenarios that are commonly used in those industries. And then the last core is what we call capacity building, and that is basically focused on training the next generation of food virologists. There aren't very many of us. And also bringing capacity to state and federal food and agriculture laboratories so that they can begin screening for norovirus contamination and sort of have it more on their radar from a public health perspective. It also includes building some educational modules at both the graduate and undergraduate levels. So that's our project in a quick nutshell, and we've been in business for about a year. So we're up and running. We have we have a website. If you go to norcor.com, you can see what we're doing, and we hope to really beef up the materials that we're putting out there for all of our stakeholders over the next year.
Well, Dr. Jaykus, you certainly have an amazing ability in your localization of the issue, and now that you've been funded and you have these other organizations supporting you, we wish you the best.
Thank you very much, and thanks for the opportunity to talk about this important food safety issue.
Well, I'd like to, if you could, in the next few minutes, to encapsulate your advice for food production, food safety, food quality, and food security managers in your closing remarks.
Okay, to summarize then, I would say noroviruses are a significant cause of foodborne disease. They need to be recognized as such. They behave in foods and the environment differently than bacterial agents of foodborne disease, but they're notable in their high degree of resistance to environmental conditions, as well as many of the commonly used bacterial inactivation strategies in the food sector. And they also are quite environmentally persistent. In terms of the fresh produce industry, we need to make sure that we understand that they are transmitted by the fecal matter of infected individuals, and also potentially by vomiting of infected individuals. And knowing that ultimate source of viral contamination then can advise us on on what we should be looking for in our facilities or in our processes as we seek to manage the problem. As it currently stands in our current world, attention to assuring that human fecal matter does not make it into the food chain is central. From the standpoint of fresh produce, that means making sure people don't defecate on fields, and assuring that the quality of the irrigation water or any other water that's used to treat crops, whether it's for fungicides, et cetera, is free of human fecal contamination. Central also to this is the assurance of adequate hand hygiene, which basically means really good hand washing, because we can't rely on alcohol-based hand disinfectants to inactivate noroviruses. It also means careful attention to illness status of food workers to assure that people with active acute gastroenteritis infections are not in food production or processing or preparation environments. In other words, they should be excluded from work. And stay tuned for hopefully in the next 4 to 5 years some substantial advances that may well help us to better manage norovirus contamination in this sector, as well as the other important food production and processing sectors that are at risk for virus contamination.
Dr. Jaykus, I'd like to extend my sincere appreciation for your wonderful conversation this morning and allow you to have a great day.
Well, you too, and I appreciate everybody listening.
Thank you very much. Have a great day.
Okay.
Bye now.
You too. Bye.